our topic is second line of defense. As we were done with the first line of defense, our topic is second line of defense. What is second line of defense? So when the first line, the bacteria passes the first line, so our second line of defense is being activated. Okay, as we know that I'm making here the first line of defense. For example, this is the first line of defense that contain the physical barrier and biochemical or physiological barrier. So first line of defense contain the physical barrier and physiological barrier, as we were done with these. Okay, so now it is non-specific, means it will not work on the targeted organ, it can work on every pathogen. Okay, so now here we will be talking about the second line of defense in detail. Second line of defense contain what will be happening in the second line of defense. So there will be killer cells and monocytes. Killer cells are basically monocytes. Monocytes are what are they? Monocytes are the type of leukocyte. And cytokinesis, that will be discussing detail in later. Fever, here when the bacteria will be passing across the first line of defense, so the first line of defense will be activating the second line of defense. So in second line of defense, what symptoms will be occurring? Fever, inflammation, means swelling, and cytokinesis. As I told you that what is cytokinesis, it is a protective protein. Active protein. Okay, it is a protective protein, and the major component of the second line of defense, we will see that as well, and as well as neutrophils. So here we will discussing about these all. So the major component of the for second line of defense are major components are killer cells, number second, fever and inflammation. Here one is missing, that is cytokinesis. So cytokinesis is not very important, but this is just like a protective protein. So now the killer cell is categorized. Into two types: monocytes and that one, I'll tell that in this video. Yeah, dendritic cells. So these both will be discussing here in this video. So what is monocyte? Monocyte is a type of leukocyte. It is a type of leukocyte. Okay, so yeah, monocytes are the type of the leukocytes. Okay, so if we talk about RBC, WBC, so monocyte will make 2 to 8% of the WBC. Okay, WBC 2 to 8%, and they are a granulocyte. Sorry, they are a granulocytes. What is a granulocytes? So it is the pigmented molecule that are absent in the nucleus of monocyte. It is a pigmented molecule. Okay, it is a pigmented molecule that is absent in monocytes nucleus. Okay, so they are, that is why known as agranulocytes. A means absence of what? Absence of pigmented molecule. Okay, nucleus. So the next point is they are originated, means they start from the melanocyte stem cell, and but they are present, they originate, they start their origination from the melanocyte stem cells, but they are present where? In bone marrow. Okay, so present means the location is bone marrow, but where they are made? They are made in, here I am talking about the monocytes. Dendritic cell, we will discuss that later. So they are made in, or you can say originate in myeloid stem cells. These are a type of cells. Okay, but you need to keep one thing in mind, that after bone marrow, they are present in bone marrow, but they stay in blood, and where they will mature. So they will stay after they made in the myeloid stem cells, but they are present in bone marrow. After that, they will stay in blood, and where they will mature. So yeah, they will mature in blood. Maturation in blood. Where they are present? They are present in bone marrow. Okay, what? Monocytes are present in bone marrow. They are made in myeloid stem cell, but where they will mature, means where they will grow or mature, kahan pe honge? So they will mature in blood. Okay, and after the maturation, their name will be changed. What they will be named? They will be named as macrophagocytes. What is macrophagocytes? So they are the cells that releases the signal, I'm gonna repeat that again, that here we are talking about the major component, second line of defense. The major components are killer cells. Killer cells type is monocytes and dendritic. Monocytes is a type of leukocyte that can be leukocyte means white blood cells. Leukocytes, the other name of leukocyte is white blood cells. So 2 to 8% of the white blood cell is made up of monocytes. So they are granulocytes, because they do not have the pigment molecule. Their pigment molecules is absent in the nucleus. And where they are present means monocytes, where they are present in the bone marrow, but made in myelocyte stem cells, and after they are mature in blood, and where they are named as macrophagocytes. Sorry, these names are very difficult. Macrophagocytes. They are after the maturation, they are named as macrophagocytes. If we decode it, so macro means big, and phagocytes means eater. Okay, so yeah, here macrophagocytes. Okay, so as I told you, they are mature in the blood. So after that, they can move anywhere, in every location. They can be in any tissue or in any organ. It can be what? Macrophagocytes can be in every, not any location. It's the quality, or you can say it's the characteristic of the macrophagocytes, that after maturation they are into any location they want. They can go in the lymphatic tissue, they can go in every vital organ. Okay, so I am writing that can go in any location can be lymphatic tissue and or any organ. Okay, so talking about the macro phagocyte, so they are the largest cell among all the leukocyte. What they are? They are the largest cell among all the leukocytes. We'll see the function of it later. So they are the largest cell of all the leukocytes. So now we are going to discuss about phagocytes and phagocytosis. So what are phagocytes? Monocytes, macrophagocytes are known as phagocytes. What is phagocytes? So we'll discuss that. Like phagocyte means engulf, they engulf. Macro and monocyte will engulf the bacteria. So by which process? They will engulf it by the phagocytosis. What is phagocytosis? Phagocytosis is the process of the engulfing of pathogens. What? Engulfing of pathogens. Okay, engulfing of pathogens. So phagocytes, I hope that is clear. Now if I talk about the structure of the macrophagocytes, so the structure of macrophagocytes is they have horseshoe shaped nucleus. Okay, their lifespan is like their life period or their lifespan is month. It can be month or years, and it can depend on the need of the body. Okay, how much how amount or how many times or till when your body needs these. So the lifespan of the macrophagocytes are depend on the body. So at a time, they have the quality. At the time they can engulf 100 pathogens. What? They can engulf 100 pathogens at a time. This is the quality of macrophagocyte. So what macrophagocytes will do? They will protect our vital organ. What are vital organs? So as I told you that macrophagocytes can go in any location or in any organ, lymphatic tissue, any organ. So what they will do? They will work as a protection. They will work as a protection for our vital organs. Vital organ means the organs by which we cannot survive. Okay, if I would ask you that eye is a vital organ or not, so of course eye is not a vital organ. We can survive without eye, but survival is possible, but we do not require it at a very essential. It's not essential need, but as compared to your heart, as compared to your lungs, as compared to your nervous system. So the comparison between organ and vital organ was that. So what macrophagocytes will do, they will protect our vital organ. Okay, so like macrophagocytes are present in organs, and they have the name according to that organ. Means if they are present in lungs, so they will have they will named as alveola. Okay, if they are present in what do we say, nervous system, so they will call as microglia or microglia. Okay, if they are present in the liver, so they will call Kupffer cells. If they are present in liver, so they will call Kupffer cells. Okay, so macrophagocytes are named according to the organ. So now let's see how they will work. So macrophagocyte produces nitric acid. What they produces nitric acid. Okay, so what nitric acid will do, it will engulf the bacteria. Okay, it will engulf the bacteria and attach to the cell membrane. Engulf the bacteria and attach with the cell membrane of macrophagocytes. So how they will work, they will produce nitric acid and they engulf the bacteria. Then that bacteria will attach, they will allow the bacteria to attach on the cell wall of macrophagocyte. Where they will be present? Will be present on the cell membrane of macrophagocyte. So now here MHCI will produce what? MHCI. I actually forget what's the abbreviation. I'll give the name of the abbreviation in the description so you can check. MHCI will made what they will made? They will made the T cells. What are T cells? We have already studied that in the first line of defense, the T cells. So what macrophagocyte attach the bacteria to the cell wall with its cell wall, and what they're gonna produce? They're gonna produce MHCI. Sorry, that will make the T cell activate. What they're gonna make? For example, if a bacteria is coming to the macrophagocytes, so macrophagocyte will attach that bacteria and MHCI will activate the T cell. So what it will do, MHCI, it will activate the T cell. That bacteria is coming, please be active and make the antibody. What was the work of T cell? It will make the antibody. So MHCI here is for the purpose, not for any doing anything entertainment or anything. It is for the purpose to activate the T cells. Okay, so T cells, yes, T cells. So what it will gonna do, made T cells to activate it for the formation of antibodies. Okay, so now by the condition you can say that this response is the specific immune response. Why? Because we are like our macrophagocytes are working on a specific target. So this response, the result of this response will be specific immune response. Specific immune response. Here will be the secretion. Here also what macrophagocyte, I'm sorry for what I'm pronouncing. So for macrophagocytes also secretes interleukin 1. Interleukin 1, it will secrete interleukin 1. What is interleukin 1? It is basically glycoprotein. Okay, so yes, interleukin work, we'll discuss that. So what it gonna do? So after then the response, the interleukin will come and the response will be generated, and the response will be going to hypothalamus. What is hypothalamus? It is a part of brain, part of forebrain. You are having diencephalon and thalamus, hypothalamus, epithalamus. So hypothalamus is a part of forebrain. What's the purpose of it? Homeostasis. It controls the homeostasis. We know that. Sorry if the spell is wrong. Homeostasis. So the response will be going to hypothalamus. So what hypothalamus will go in the result? Hypothalamus will be rising the temperature. Result will be the rise of temperature, because all this happening, macrophagocytes like bacteria is coming, antibodies present, so there must be a response. Your body will be creating a response. So what hypothalamus will result in the form of the rising of the temperature, and what our inner body will do? So when the temperature will be rise, all pathogens will be killed. Pathogen will be killed. Okay, so all these pathogen will be killed. And why they will be killed? Because we know that enzyme will be denatured. So at in the rise of the temperature, the enzyme will be denatured. We know that when enzyme will work on the 37 degrees Celsius, and enzyme will work always in the optimum temperature. So the temperature will be rise if in the body, so pathogen will be killed and enzyme will be denatured. Because the temperature rise, so the pathogen will be die, because the pathogen are made up of enzymes. Okay, so enzymes will be denatured. Okay, so this was the process. Like if the enzyme will be denatured, so automatically pathogens will be die. This was the second line of defense, macrophagocytes or monocytes. Okay.